The problem we are faced with here is the size of chemical space. Chemical space is extremely large. I'm not going to attempt to describe for you how large it is. I'm going to use one of my favorite authors instead, Douglas Adams. In The Hitchhiker's Guide to the Galaxy, Adams said, "Space is big, really big. You just won't believe how vastly, hugely, mind-bogglingly big it is." And I think if you put the term chemical in front of that, it gives you an idea of how large chemical space can be. Quantitative estimates vary dramatically from what looks like a very low number of around 340 billion to a number of molecules that may be more than the number of atoms in the universe. So somewhere along that scale is the number of accessible molecules, and it is enormous. We will never, of course, be able to get to searching these complete spaces, but the spaces of molecules that we have available even at the moment are very large. One I'll be using extensively today is the enamine real database. This is a database of high probability of synthesis molecules from enamine. It's around 1.5 billion molecules that's focused firmly in drug-like property space. So here looking at polar surface area in log P, you see the distribution firmly centered in sort of traditional rule of 5 drug-like space. The enamine collection is also very diverse. Here on the graph, the x-axis shows the number of Bemis-Merkel cores, and the y-axis the number of sublibraries in enamine that have those Bemis-Merkel cores. A Bemis-Merkel core is the image on the right side, and the parent molecule for that core is shown on the left. So the real collection contains a great level of chemical diversity, focused mostly in traditional drug-like space. And we will be searching this prospectively to give some in-process results for some of our tools toward the latter end of this talk. Now, of course, 1.5 billion is a very tiny fraction of the available chemical space. And we've been collaborating with AstraZeneca to look at fractions of their available chemical space. Their estimates say that their virtual library space is about as large as 10 to the 15. And of course, searching that entire space is going to be very challenging. But they're very interested to understand what advantages accrue to testing, to screening larger and larger fractions of this 10 to the 15 space. So I will be looking at some work we've very recently published with them. The paper came out just this week. So when we at OpenEye are thinking about developing tools for searching spaces of this size and larger, we focus primarily on two fundamental molecular properties: molecular shape and molecular electrostatic potential. And the reason we do this is twofold. Firstly, they are fundamental molecular properties. Molecules just have a shape and they have an electrostatic potential. But also, these two properties are key to understanding probably the most important event in drug design, protein-ligand binding. Good protein-ligand binding only occurs if there's complementarity in shape and electrostatics between the ligand and the active site of the protein. So we can use this lens of shape and electrostatic properties to think about designing molecules that fit the on-target active site well and give us potent molecules. But we can also think about using the same lens of shape and electrostatics to design molecules that do not fit well to side targets or off-target proteins, thereby addressing problems not only of potency, but also selectivity and side effects. Today I'm going to focus on the first of these and use shape-based methods to discover new, interesting, biologically active molecules. The computation I'm going to do today is all going to be done inside our cloud platform called Orion. And I'm going to focus primarily on two aspects of Orion: a way to develop customized workflows suitable for screening at these very large scales, and to provide the compute background to execute these calculations efficiently and quickly. I'm not going to spend much time on the analysis and the sharing, but the results that we've generated have been analyzed and collaborated on inside Orion. Our goal with Orion is to provide all of the common computer-aided drug design CAD tools through probably the most common information gathering interface of the 21st century, the web browser. So our goal is to democratize the process of molecular design and put both the compute power and the tools in the hands of anybody who has the inclination to do it and access to the web. So today our specific goal with Orion is virtual screening, and the way we conduct virtual screening at OpenEye is illustrated schematically here. It's a three-part process. Firstly, preparation of molecules, things like application of protonation state and tautomar state. Secondly, we generate conformations as a pre-step. And this allows us to amortize this relatively compute-intensive step across all subsequent searches. It also accelerates subsequent searches because they're performed rigidly. The subsequent searches are of two kinds, either ligand-based, looking at shape or electrostatic feature similarity, or complementarity-based, structure-based, or docking tools looking at complementarity in shape and chemical features between ligand and binding site in the protein.